welcome and thank you for joining us today. I'll shortly introduce my co-chair, Dr. Dan Bryson, a research fellow at the Developmental Neurosciences Department at University College London, who will introduce today's speakers. But before we get started, I'd just like to go over the background to the session. The Physiology of Obesity, From Mechanism to Medicine series started a few years ago when myself and fellow co-organizer Dr. Peter Aldiss at the University of Copenhagen felt there was scope to develop an early career researcher focused symposia that could give more speaking opportunities to early career researchers and bring those of us in obesity and metabolic disease together to network and disseminate results. After 3 successful symposia, firstly at the European Congress on Obesity, and then Experimental Biology, and a satellite meeting at Physiology 2019, we are now delighted to bring this webinar series to the Physiological Society and give early career researchers the opportunity to present at a time when opportunities are limited to do so. So far, as this series draws to a close, we've had over 2,000 registrations across more than 40 countries and over 2,500 views on YouTube. And it's great to see so many of you with us for this final webinar, which is a change from our usual format. Today, we have 2 leading experts in physiology, and this will give you the opportunity to quiz them about the field, their research, and how you can take part in this arena. Both of our speakers today will give 15-minute presentations, and then we'll have time for Q&A at the end. You'll be able to submit questions through the Q&A button at the bottom of your screen, and then my co-chair and I will direct these questions towards the speakers. These questions can be submitted anonymously. However, please provide your name and institution if you're comfortable with those being read out. We may not have time for all of the questions, but we will try and get through as many as possible. You can upvote these questions and also type your own answers or comments to other attendees' questions. And please note that this webinar is being recorded today and will be subsequently available to rewatch via the Society's YouTube channel. All that remains for me to say is to thank the speakers today, the Physiological Society, my fellow co-chair Dan, and Dan, it's over to you to introduce the speakers. Thank you.
So good afternoon, everybody. I'd like to thank Joe and Pete for inviting me to co-chair this session. I think the original idea was for Pete to co-chair this session, but he's been otherwise detained, which is a real shame because of the hard work that Joe and Pete have really put into this event. I think there's a bit of a long reason for why Pete can't join, but if you follow him on Twitter, I think you get a pretty hefty rundown of why he can't be here. I just want to sort of say, you know, as an early career researcher, so I'm actually becoming a trustee of the Society as an early career researcher. And the idea is that I'm there looking after the interests of the early career researchers at the Physiological Society and to champion early career researchers. And this event has just been a phenomenal success. And so I'd like to congratulate Joe and Pete on that. And it just shows what any of you that are listening along today can get involved with at the Society. If you've got anything that you'd like to share, any ideas, any concerns, then I'm all ears. So let's progress. So the first speaker today is Juleen Zierath, who is the Executive Director of the Novo Nordisk Foundation Center for Basic Metabolic Research at the University of Copenhagen, Denmark, and leads the Integrative Physiology research groups at both the CBMR and the Karolinska Institute in Sweden. Juleen is a dedicated physiologist whose research investigates the interactions between circadian rhythm, diet, and exercise to identify targets for the prevention and treatment of metabolic disease. And I actually had the pleasure of seeing Juleen deliver the President's Lecture at Europhysiology 2018. So I know we're in for a real treat. So please take it away, Juleen.
Can you see the slides all right? All right, so this is great. It's a real honor for me to be able to, join this series. And I thought for the 10 to 15 minutes that I had, what I wanted to do was just give a perspective from my own career background and kind of how I got into science and how some of the experiences I had early in my career got me to the point of where I am today. And that also impacted the kinds of research questions that I have been interested in. So I'm from Wisconsin originally. And, I never would have imagined that I would have ended up in Sweden ever in my life when I was even thinking about my undergraduate degree. And my undergrad degree was at a state college at the University of Wisconsin at River Falls. And I was a first generation student. No one in my family had ever gone to a university before. And so in that respect, I probably got little guidance in terms of career planning and career development, full stop. But I was interested in athletics and sports. And at the University of Wisconsin River Falls, I was on 3 sports. I was competing at a time when women could be in 3 sports in a year. And I was really interested in athletic performance. And I had an undergraduate degree in physical education and business administration, actually. And that's a strange combination, but my father always said, Julie, take business, you're always going to need business. And in fact, I used that education and training a lot, because if you think about writing grants, you have to write a budget, you have to manage a team, you have to manage your economy. So surprisingly, that part of my education was more valuable than I ever would have expected. But my undergraduate degree was, it was a teaching degree. And by the time I finished, I was certified for teaching in high schools. But I was so interested in athletic performance and just sort of the marvel at how the body responds to different training paradigms. I knew I wanted to know more. And so I followed, obviously in all the textbooks, Per Olof Ostrand's work. And, with every page, I was just more amazed with muscle physiology, how the body adapts to different environmental stressors. How is it that people can run a marathon in extraordinary times? And the other was health and prevention. And on a personal narrative, my grandfather had a bypass surgery. And one of the first lines of treatment was cardiac rehabilitation and exercise therapy. So that really fueled me to say I wanted to know more. And I moved on to Ball State University, where I had been reading a lot of the work of David Kostal. And I mean, besides being an extraordinary writer, he could really explain things in a very accessible way. He was studying all the things that I was really interested in. And when I went to Ball State, I did my master's under the supervision of Ann Snyder. And at the time, what the project I had was really to look at more nutritional aspects of distance runners and more focused on the female athlete. And we were doing some studies on bone density and how bone is adapting during physical exercise and training. And I had the opportunity to work with Bruce Craig. And Bruce had been trained by John Halazzi, who I'll get to in a minute. But we were really looking at exercise, and together with John Hawley and Daryl Newfer and Mark Sleeper at the time, we were looking at how different modes of exercise, swimming or running, in my case, affected bone density. In John Hawley's case, it was muscle enzymes, muscle metabolism, and Mark Sleeper was looking at adipocyte biology. But when I finished my master's, I really had kind of two paths. One was to get a PhD, and the other was to pursue a position as a research technician. Because at the same time I was interested in doing a PhD, I wasn't quite sure what I wanted to invest my time in for a PhD work. And kind of in an interesting story that I can just share a little bit more, I had applied to one opportunity, and I was listed second for the job. And the minute I got the notice that I was listed second for the job, I got another call, and that was from John Halazzi. And he invited me to come to St. Louis. And I worked in his laboratory for about 3 years. And that was a phenomenal time, because this was before the glucose transporters were cloned. We knew very little beyond insulin receptor signaling. We knew there was a receptor, but beyond that, there was question marks. And John had a phenomenal environment where he had a downstairs lab where it was more on mechanisms of mechanisms by which exercise and muscle contraction could steer metabolism at a more cellular level. And he had an upstairs lab, which was his applied physiology lab, where he was performing studies in people with diabetes or obesity and asking how he could translate what we learned from the downstairs lab, the mechanisms, to the upstairs lab, which was the application to people who are having these metabolic disorders. And so a lot of the work that we did when I was in John's lab was understanding how exercise and cell stressors affect glucose metabolism. And I think one of the big things that we kind of contributed to was that exercise increased glucose uptake by an insulin independent mechanism. And so after the time of the work in John's lab, I knew I wanted to do a PhD, and I wanted to focus on muscle metabolism and trying to elucidate some of the signaling mechanisms by which insulin or contraction steer metabolism, and how exercise can be used as a medicine to, prevent or delay insulin resistance. So then I went on and did a PhD at the Karolinska Institute. And my work there was metabolic regulation of glucose transport. And that work actually is published in a supplement to Active Physiological Scandinavia. And after that, I went to Harvard. I did a postdoc with Barbara Kahn, where I was working more on signal transduction and adipocyte biology. And in 1996, I moved back to Sweden. I was funded from the Research Council and started my own group there. I guess the message here would be that I never would have carved out this career path from the beginning. I probably had no idea that I would have been able to work across the ocean. And what steered that for me was just the interest in the scientific questions I had, the networking opportunities that I made along the way, and to some extent, not having a lot of fear, because when I moved to Sweden the very first time in 1989, I had never been out of the country. And so I had some kind of conviction that I really definitely wanted to study these things. I wanted to resolve these questions, and trusted that the science would take me there. So today my group is really interested in understanding not just one molecule, but how signal transduction and transcriptional regulation is orchestrated in muscle cells, and how this is influenced with insulin resistance or obesity, and how exercise can rewire a muscle, and how muscle communicates with other organs. And probably the most recent area of science from my team is understanding the physiology of how our body is aligned with our circadian clocks, and how we can fine tune our metabolism through different periods of day. And so, would there be the best time of day or a better time of the day to exercise, or is fasting beneficial for our metabolism? How do we synchronize our exercise and our nutrient interventions to our molecular clock? And is there a way to fine tune metabolism to improve mitochondrial function, insulin sensitivity, and glucose metabolism? And so with this kind of brief introduction, I can say that I'm working between two environments now. I'm at the Karolinska Institute, where I have a research team, and I'm also in Copenhagen, where I'm running a research institute with over 200 scientists that are really focused on metabolism. But Joe, I wanted to give this Dan, I wanted to give this as a brief introduction of what I'm doing and how I got here. And I hope that we have time for more questions on career development and career opportunities.
Thank you very much, Juleen. That was a lovely overview, and I'm sure that many people will have I've already got a few questions as well lined up. So yeah, very, very much looking forward to posing those later on. Thank you very much. Okay, so let's then move on to the next speaker. So the next speaker is Mark Reitman. So Mark is a senior investigator in diabetes, endocrinology, and obesity branch chief at the National Institute of Diabetes and Digestive Kidney Diseases at the National Institutes of Health in the USA. He's interested in the mechanics and translational understanding of diabetes, energy homeostasis, and obesity. And so Mark, whose research has focused on the physiology of obesity, has leveraged mouse genetics and pharmacology to understand the regulation of metabolic rate and body temperature, with a focus on the bombesin receptor subtype 3 to develop candidate treatments for human obesity. Okay, so without further ado, if you'd like to attempt to share your screen, Mark, we'd love to hear from you.
How's that? Is that working?
That's wonderful. Take it away.
Okay, well, first of all, Joe, thank you, and Dan, thank you very much for this opportunity to talk a little bit. I am used to talking about my career informally, but to do it formally is not my usual thing. So hopefully this will be useful to people. And if you do have any questions, I really do enjoy providing suggestions and advice and interacting with earlier stage people. And my email is on this slide, mark.reitman@nih.gov. I have no disclosures. Okay, so I thought I have a slightly different style than Juleen. What I will do is just give you a brief outline of my career path. I obtained an MD PhD. I was an undergrad at MIT. I did an MD PhD at Washington University. My PhD was with Stuart Kornfeld, and there I studied the biochemistry of glycosylation, and in doing so, figured out the enzymatic defect in a couple of inborn errors of lysosomal biogenesis. And I really liked that and thought I might want to be a geneticist. And I'll talk about that on the next slide about how I decided I didn't want to do that. I then trained clinically in internal medicine and endocrinology and metabolism and became fully qualified in those. And then I went to the NIH where I did a postdoc, where it's a very basic lab looking at chromatin structure, chromatin regulation using chicken globin genes as the model system. And then I was recruited to stay at the NIH, where I became faculty, initially continuing to work on globin gene expression. And then with the discovery of leptin in 1994, I decided I would study that and ended up really diving full force into understanding adipose tissue and then obesity. I got Wanderlust, and in 2002, I went to Merck, where Merck Sharp and Dohme, ex-US, and there I was one of the leaders of the obesity drug discovery and drug development effort. I stayed there 9 years, and then I was recruited back to the NIH, where I have a small lab and I run a department, which is sort of like an endocrine division within a medical school. So that's the bare bones of what I've done. And now I thought I'd talk a little bit about the decisions. So when I was asked to do this, I was given two questions. One was, how has physiology shaped my research? And so I thought I'd give two underlying principles that I think have governed my path. The first is that I think of science as the understanding of mechanism in nature. That's how I define science. And I think that the science of humans is physiology, how it works, how it doesn't work. And certainly the most important and interesting to me model system is humans. So where did I start? I started my PhD doing inborn errors. This is before the genome. This is before genetics existed as a clinical discipline. And I thought I wanted to be an inborn error geneticist until I did pediatrics clinically. And I realized I just didn't have a personality to be a pediatrician. I like adults and older people, but trying to be a doctor for kids was hard for me. So I decided that inborn errors was no longer where I was going to go. So then I did a clinical training, and I was so busy, I really hadn't had time to figure out exactly what I wanted to do with my life. And so when I was offered a clinical fellowship at the NIH, part of that offer was that I could choose whatever lab I wanted for a postdoc. And again, I had troubles making that decision and basically chose what I thought was the best lab around. It was run by a guy named Gary Felsenfeld, who was a National Academy member. And so I decided I'd studied chromatin globin gene expression for my postdoc. And that went very well. And then I got recruited as a tenure track person at the NIH, and I was told I could work on whatever I wanted. And at first I continued on working on chromatin and globins, but I was in a clinical place. I'm trained as an endocrinologist, and I was looking around for what I could do that would be more endocrine related. And I decided obesity was really a backwater. And with the December 94 publication of The Discovery of Leptin by Jeff Friedman's lab, I decided with my background in transcriptional regulation, I would study the transcriptional regulation of obesity, meaning start with leptin. And we did publish an early paper on leptin and its transcriptional regulation, but within months I realized not enough was known about obesity, or how leptin worked and where it fit in the grand scheme of things to really be doing transcriptional regulation, that really you needed to understand the physiology. And about that time, there were 5 monogenic models of obesity in mice. All 5 in the early to mid 1990s were discovered at the mechanistic level: leptin, leptin receptor, melanocortin pathway genes. And so I decided the genetics was really important. But very quickly, I also decided that, again, understanding these genes still didn't tell us much about obesity. We really needed to do organismic physiology. And the place where you could do that was mice. So I really got excited and interested in using mice to study physiology, and I studied lipodystrophy. And the other decision point was, I'm really interested in trying to understand human physiology. Why am I studying mice? And there's a couple of answers to that. One is the mice are manipulable. You can do quick, defined experiments. But on the other hand, mice are not just small humans in some ways. Okay. So there's a few summary thoughts, if you will, free advice that I've tried to distill as things I generally tell earlier career people. And the first question is, how do you pick a question? And I think the way you pick a question is you try and ask the most important thing you can think of, the most impactful thing you can think of, where you have some special insight or tool or ability. I personally don't want to compete with everybody in the world. I want to do something that not everybody's doing, and hopefully that'll be important, but that's certainly where my personality works. And part of that is I really want to be interested and excited about what I work. If you're going to be working ridiculous hours, it should be fun. And the last thing I've done to an extreme degree in my career is follow the science. I've probably, to an unhealthy extent, shifted the things I've worked on many times in my career, and that's been fun for me. And it's allowed me to have a very broad knowledge base, although not necessarily as in depth in certain areas. So when you're approaching something, what sort of advice do I have? One is, you want to invest in expertise and tools. You want to be the expert in what you're doing. You want to develop tools. It may take, it used to take, a year to make a mouse and characterize it. But I was willing to do that because now you had a unique tool that sort of was a benefit of that commitment you'd made to it. The other thing is, you really want to surround yourself with smart people, hire smart people, and talk to smart people. The other thing that I think is sometimes lost in the current world is you really want to be sure your science is correct. You want to be your own worst critic. You want to think of that experiment that will disprove your own science. You don't want somebody else to. You want to be proud of how many ways you've asked, is this really the right answer to this hypothesis? And then one of the things I like to tell people is, you really want to be an early adopter of techniques just after they're worth adopting. So I call that a fast follower. You don't really want to be on the bleeding edge where you spend 3 years trying to get an assay to work. And by this I mean, in your own little field, of course you want to develop assays, but once transgenic mice works, once CRISPR-Cas9 works, once data science becomes the tool of the realm, then you want to adopt those quickly, because the advances are going to depend on using those modern tools that now allow you to ask questions you couldn't have asked before. And finally, this sort of builds on what Juleen said about business. I think things that really, when I was growing up in science, nobody told me how to run a lab. Nobody told me how to manage people. And nobody told me how to give a good seminar, how to communicate, how to give advice, how to give suggestions for improvement to people. And I think those are all really, really, really important in any aspect of your life. But certainly as you, for example, run organizations in science. So you need to communicate, you need to get good at managing people. And part of that is, and this is where I may fail, is you really want to be very, very encouraging. You want to, the more exalted your assumed position is, the more people take criticism harshly from you. So it's really important to be encouraging, but also to be honest. And you want to create an environment where everybody gives feedback. And finally, we stand on the shoulders of giants. We want to nurture the people who are the future of science. So the next question I have is, how will physiology be important to obesity research? Well, historically, we had no tools. I mean, it was sloth and gluttony was what caused obesity, because we really didn't know how to study it. And then the genetics led to monogenic obesity. And I think the take home from that is that obesity is a disease. It's amenable to the tools of biological science. And in a sense, one of the conclusions of that is obesity is a brain disease. The metabolic complications of obesity are peripheral to the brain, to the fat, to the muscle, to the liver, to the vessels, to all over. But the regulation of the food intake is in the brain and the energy expenditure. And then the next advance was using genome-wide association studies to study obesity. And I was just sure that obesity was going to be solved by that. And it turns out, no, it's hundreds of loci with thousands of variants. So it turns out obesity is one of the most complicated genetic backgrounds of any disease. And so that really has led to the need currently that we really need to understand physiology to understand the genetics. I would argue we still don't understand the physiology of obesity. And my time at Merck really convinced me that we didn't know enough human biology of obesity to be doing this in humans. And that's why I sort of reverted to the mouse biology when I left Merck and went back to the NIH and decided what I really wanted to do was understand the neuroregulation of energy metabolism, because we didn't know enough about it in order to make rational choices of enzymes or pathways to target for the treatment of obesity, and that the mice were imperfect but reasonable models, meaning that intermediary metabolism and brain organization, all that stuff is the same in mice and humans. However, when you get to really nitty-gritty details, like for example, thermal biology, mice and humans are really quite different. So where's the future? I think we're always going to need physiology to test hypotheses. You know, we're always going to need animal research. We don't have computer simulations that are good enough, because if we knew how everything worked, we wouldn't be needing to do the animal experiments. And then I think, again, where's this going in the future? You know, I'm a very mathy person. Years and years ago as an undergrad at MIT, I really enjoy math. I still think of it. And I really think one of the useful tools is data science. However, it's a hypothesis generating tool, and you're going to need physiology to follow up on that. And looking at the time, I think I've used up my time. So I was going to talk a little about thermal biology, but I'll skip that and we'll go right to the questions. Thank you very much. And I'm open for questions, or back to Joe and Dan.
Thank you, Mark. That was a very interesting talk there. So if we can start our question session. Juleen, I was delighted to hear you talk about career planning and development. Just how important is that for kind of today's undergraduates and PhD students, and also postdocs as well?
Well, I actually think that you're making a huge investment in yourself. And in that regard, I think planning is important. That being said, I don't think the plan should be so rigid that you are unaware of unique opportunities that come up. So I would really recommend that you have a sketch on where you want to get. And it's difficult to plan out 10 years. It really is hard to do that. But I think you can have a dream, and you can have a goal of where you think you want to be. And that might change. So I would say definitely get a plan, get a lot of information. Networking is hugely important. Gather up as much information as you can about the next environment that you think you want to get to. And, you know, interview people. And just take it as advice and find a way to put that list together of pros and cons for different things that you plan. But I was really clear that I knew that I wanted to have an academic position. I was very curious about science. And I tried to find the labs that I thought were the most exciting for what I wanted to do. And I was very persistent. So I think all those things are important in the mix. I don't know if that answered the question, but.
No, it did. Thank you. Because these are things that you had touched on in your talk. You talked about the importance of the scientific questions, the networking, but also the fear to go out there and pursue those, but then also having that conviction as well. And do you feel it's the combination of those which has led to your success?
I'm not sure. I can't really say what, I mean, it's hard for me to do that self-analysis, okay? But I just know that I really had a clear idea of this is the biology I'm fascinated. Mark has the same feeling. I was just fascinated about muscle physiology and wanting to know more. And so I think if you are that interested, then the next would be to follow that interest and don't be deterred by imaginary blocks. You know, and actually I think that's one good thing about moving to Sweden is that sometimes environments have sort of do's and don'ts of the environment, you know, certain ways you should behave. And when I moved to Sweden, I kind of I didn't understand that. So I just followed my conviction and I didn't pay attention to, oh, you shouldn't do this or you shouldn't do that. I just followed my bliss. I followed the things I was really interested in. So I think somewhere you got to listen to your inner voice, you know.
Okay. That's good to hear. Thank you. Mark, I was delighted to hear in your talk that you were still championing animal research, because what I found anecdotally, or at least in my experience, is this really is on the wane at the moment. Do you still feel is it being important?
So I'll answer that and I'll also expand on it. So first of all, I have a clinical side and I'm very, very worried that we're not doing enough clinical research. And so I think the role of a PhD in clinical research, the MD teaches you more normal and abnormal biology than you could ever do voluntarily. The PhD teaches you what a rigorous controlled experiment is. So I think the melding of those two disciplines is awesome. I think we're training fewer and fewer clinical researchers. So I would like to encourage PhDs to really pay attention to the clinical side and use that. Now, on the animal side is I tried to make drugs for obesity for, let's say, 9 years. And by the end of it, most big pharma had gone away from it. The drugs we have for obesity were discovered by accident. Almost nothing was rationally designed. And you know, whereas the hubris at the time I was doing this 15 years ago or 20 years ago was that, oh, we now have all these genes, we'll just make these drugs that to these genes and we'll have drugs for obesity. And that's, you know, well, with rare exceptions. If you have an endocrine deficiency, if you're missing leptin, leptin replacement is magic. If you're missing MSH, MSH replacement is magic. But you know, there's a handful of people in the world who fit those conditions. Now, rare diseases teach you about common disease, but you need that. So I think you really, really, really need the mouse research, and it goes much faster and it guides where the fruitful areas for human research, which are orders of magnitude more expensive and difficult than mouse research. Hope that answers. I expanded a little bit.
I would agree with you. Oh, can I just jump? I would agree with Mark. I think you need both. I mean, sometimes it's hard to approach mechanism in humans. And you really can actually explore things more in animal models. But then, of course, you want to have human validation as well. You want the human relevance.
Yeah, I agree entirely. Thank you. So I really like this idea of following the science as well. But in today's job market, where people are often kind of changing fields, do you really feel that that's kind of possible for the next generation?
I hope so. I've done that, and I think what it does is it cross-fertilizes. You bring ideas that others may not have just because you've thought about other things in addition. But I really think it's important. So for example, coming from humans back to mice, I really was convinced mice were small humans and we could figure out how to study obesity in mice. And recently in the vignette I was going to tell at the end of my story, that I think mice cannot do the thermal biology in mice is different than humans, and there is no exact way to compensate for it. You can do various approximations, but I really don't think mice have a usable thermoneutral zone, and yet humans live in their thermoneutral zone roughly all the time to a first approximation. So, but that again says, okay, so that's not a failure of mice. That says, okay, now we know this. We just have to be careful when we do mouse experiments that when we translate to humans, it's going to be an asterisk and we have to pay specific attention to this to make sure we don't make a mistake. But I do think people should change as best they can their careers. I think it is harder to do, but it's important.
Well, all right, so at least in Sweden, I mean, a lot of my colleagues have biotechs. And so even in Denmark, there's a, push for people to be innovative. And so that would be a great example of where one could be a real innovator. You could start a biotech, you could move into that sphere. You could move back into an academic environment as well. So I think people are less hung up on career tracks, and once you're on it, you can't get off it. I think we start to see people getting benefits from many different kinds of environments, and seeing the value of that in tackling these huge issues that we're dealing with, obesity and diabetes.
And I think that's something, if I might interject, that's something that the UKRI, which is the UK Research and Innovation, the new head of that, Juleen has recently come out and said she wants people to cross-fertilize. She wants people to cross-tracks. She wants a huge network of people coming from different environments. And so I think, whereas previously people were, and I'm guilty of this as well, of seeing people go straight through the academic track, never leaving it. I think the future is going to be encouraged for people to do that, and it's something I think that early career researchers should be definitely thinking about.
Mark and I have that in common. I mean, I worked at Pharmacia in a biotech called BioVitrum, and I was the head of target discovery and validation, and came back into the academic environment. Mark did the same thing. So I think people are less hung up on you're on a road, you have to stay on it.
And I think the thing that industry allows you or requires of you is that you have to be very flexible about what you work on. In industry in particular, a target can be decided, and at least at Merck where I was, I thought generally very intelligently and transparently that we now have extra information. This is less interesting or less likely to be a success than target B. So we're going to drop target A and move to target B. And that requires extreme flexibility to do. And I think that's good. And one other point is that this going from academe to industry and back, when I was a grad student, it was like, oh my God, industry is where people went who couldn't make it in academe. And then you have the Biogens and Genentechs and the brightest and best people go to industry. And it is somewhat of a two-way road. I mean, I left NIH, went to industry and came back to NIH. I think it's sometimes harder to come back to academe because you don't bring grants with you. So that's a problem, but at least at the NIH, you didn't have to bring the grant, and they knew me and they really wanted me back.
Yeah, and I think going forward, it will be much easier for people to come back, especially if we're going on the track that UKRI have set forward for us. So that's marvelous. I think that perhaps we can go to the floor now, if it's okay with everybody else. So we've got some questions that have been posed by keen listeners. And so the first one is from somebody called Wouter Peters. It doesn't say where they're from, but it reads, "Hi, Juleen. Many thanks for sharing your career path. We all admire what you've done for the field." And then poses perhaps a personal question. So as moving from an English speaking country to a country where they speak a non-familiar language, how did you find this transition? Did you experience it as an obstacle, or were you able to pick up the language quickly, even though English is the universal language of science?
All right, so moving, I moved to Sweden in 1989. It was a very different time. Now I feel like the world is more harmonized in terms of language. We didn't have internet, we didn't have email, we didn't have any of that when I moved. But at least with the science, both in Sweden and Denmark, everything is in English. So when it came to my work, that was never an issue because absolutely everything was in English. The place where it was more challenging was on a personal level. When you started to meet groups of people, large groups of people, then everything went to Danish or Swedish, and that obviously you feel a little more left out. But you do learn the language after a period of time if you're in an environment long enough. So it hasn't been a huge hurdle, to be honest, especially now when everything is just translated.
Yeah, I worked in Germany for a year during my undergrad degree, and it was a similar situation. Everyone spoke German in the lab, but you get out into the social centres and everyone's speaking German. And I was only there for 1 year, but I felt like I did a decent job of the language, and I left conversational at least, and that was I felt like that was a great sort of skill for me to pick up as well. So not only do you go and learn some great science, but you also learn a new language, which I think is just such a bonus. So let's move on with the questions from the audience. So we have a question now from Magnolia Aritza Nieto, who's a scientist and entrepreneur. Exciting. So she's a founder and CEO of EpiWell in Ithaca, NY, I'm presuming that's New York. In adults, the response to diet and physical activity is personalized. In our study, we are able to identify unique cases in our population. The rodent epigenome is very different than the human epigenome. I think I have a case of human genetics. However, I'm not sure how to move it forward with this case. What would you recommend a small entrepreneur group on how to approach these kinds of cohorts that could help understanding the cases that they're finding? I mean, that one is for either of you, I guess. So if either of you have any comments on that, then please bring forward. I think what they're trying to say is the differences in between the human and the rodent, and that they.
I think also, Dan, it might be a slightly larger question in that for any kind of entrepreneurs out there, are they able to approach university groups who may be able to give them insights into the data sets that they are collecting? Do either of you have any thoughts on that?
So I work at the National Institutes of Health, and I think where the NIH Clinical Center really shines is in studying small groups of people with rare diseases and using that knowledge from those rare diseases to expand our knowledge base to see how it applies to more common forms, the genetic obesities informing the common obesities, for example. So I don't exactly understand what the question is for this, but I think clearly obesity will be a personalized medicine situation, but personalized medicine works best when you have lots of different treatments that you can filter to the right patients. And at Merck, I was trying very hard to figure out how you'd approach personalized medicine. And we don't have, dozens of treatments for obesity that all work, but only in a small subset. So we have very few treatments for obesity. And so I think personalized medicine is much harder. You have to have discrete treatments that apply to subgroups, and I don't think we have a lot of those treatments to apply. So I think it's a very important area for the future, but we're not there yet. I hope that's not too depressing.
A lot of it's going to be very deep phenotyping, and I mean, you're treating the phenotype more so than you're treating the epigenome, I think. A good example of that with diabetes is Leif Group has started to have subsets of diabetic people based on the phenotype, and that will help inform the treatments.
Thank you very much both to that answer. So I would like to ask something which it's kind of a follow-up, but not necessarily. So thinking about treatments for obesity and looking into mechanisms of obesity, and for me there are some sort of striking parallels with the field that I'm from, which is cardiovascular disease. And one of the questions that I've quite provocatively asked some esteemed members of our community in the past is, we know a lot about what happens to the body, but it just seems that this knowledge that we have isn't necessarily filtering through to populations. It's not being properly applied. And, we put enormous amounts of funding into research for these. And one question I often like to think about is, would this perhaps be this money better spent on applications to get into population? So community groups, exercise community groups, healthy eating community groups, for example. And I think this applies across both, metabolic physiology and cardiovascular disease, rather than maybe just focusing on disease mechanisms all the time. And, bringing in social psychologists and things like that to our research threads to try and push the field forward and try and have a real impact on society. Either of you can answer that question or respond to that comment.
I think you need a little bit of both, though. I mean, one of the things I did in a summer internship was work with Art Weltman in Peoria, Illinois, at St. Francis Hospital. And they were putting a lot of resources into cardiac rehabilitation and that, you know, and adult fitness programs. So I think you want evidence-based recommendations. And the only way you're going to get that is by research. I mean, Mark was, in his introduction words, was talking about the benefits of a PhD is that you basically have rigor and you have some kind of standing on the advices that you're giving to people. And so I think you really need that. Otherwise, to some extent, you're gonna have a lot of quackery, right? And we've seen that with a lot of diets. You know, diets are prescribed, but there's no evidence that would say only eating bananas is gonna make you lose weight. I mean, so I think one needs to use both. And I do think there is a responsibility that researchers should have for society in terms of finding a way to bridge what we're doing in the lab to communicating that to people, because people are hungry for knowledge. People are fascinated by the kinds of research that we're doing. And who better than us to be able to explain that to, you know, the community and get them excited about it as well. And the other side of this is that more and more we're learning that we can learn from citizens. I mean, we can learn a lot from people with diabetes. We need to have a relationship where if we're going to study obesity or diabetes, it's important to actually bring people with obesity or people with diabetes on board to help inform some of the questions that we're asking so that they are clinically relevant. So that's just my 2 cents worth on this.
Yeah, I think it's really important to have a diversity of approaches because, for example, epidemiology is a really different science than, you know, transcription regulation, but they're all really important. You need to have all these things and then cross fertilize. Now, a basic science might refer to epidemiology as hypothesis generating. That's fine. You need hypothesis to then test rigorously and in a different setting to see how it applies. And the other thing is, I think Juleen was saying that we really have to communicate because, for example, in obesity, exercise is not a cure to obesity, right? If you want to lose body kilos, the best way to do it is lowering your food intake. That being said, exercise is a really, really good way to get metabolically healthy. And obesity and metabolic health are only somewhat correlated. You can be quite obese, at least by BMI, and be incredibly healthy by exercising. And that's one point. The second point I want to make is that newspapers, you know, news is by definition something new. And every time somebody thinks of a diet, that's new and news. And it's very frustrating to science where, you know, to a rough first approximation, a calorie is a calorie. You know, I know people don't want to hear that, but the diet that works is the one you stay on for the rest of your life. Exercise is good for everybody, not just obese people. These are truisms that haven't changed, been known for, you know, I was just in preparing for this, didn't show it, but I was looking back at Osler's 1892 first real textbook of medicine. And he basically says, you know, diet and exercise are what you need to do and genetics count. I mean, not quite those words, but he said those things. So things haven't changed and we just need to figure them out better. And the other thing is, I don't have a public health politician's personality. But in reality, I think if I wanted to do the most I could for the world of obesity, I'd become a politician and figure out how to spread the word of healthy social living. How do you build cities where you have to walk? How do you encourage exercise? How do you do all those things? And I think that's an important thing for scientists to do, is to make it easier for people who want to do that to do it correctly and rigorously.
Thank you for that, Mark. That was yeah, and Juleen, they were fantastic answers. I'm gonna throw back to Joe now, just in case he's got anything he wants to.
Yeah, there were a few more questions which came in through the chat. Luke McNever, a PhD candidate at the University of Victoria, was interested in what are the important things to look for when selecting a lab for a postdoc. And secondly, as PIs, what do you look for when hiring postdocs? Julian,
I can take the part. Yeah, in terms of looking for a lab, I would say, presumably you've completed a PhD or you're about to finish the PhD. So I would say, obviously, that's what you have in your hands. And I would say when you look for a lab, it would be one idea to try to find an environment that will either complement what you have. I mean, you don't want to go to a lab that does exactly what you're doing, because I think you're not going to learn as much. And if you're working in obesity, sometimes it's a huge, huge leap to go to a postdoc in infectious diseases or cancer. So I would say probably find an environment where your skills and what you've acquired is going to be a strength in that environment. But at the same time, you're going to learn something very new that will make you quite different than the person that you trained with earlier. And it will make you very different than the person that you're training with on that day. So that in the next step, when you go off and build your own career, you've sort of accumulated experiences that make you somewhat unique. And that will allow you to differentiate yourself from all the other people around you. So I would say that it's important to complement, but also find something that will make you really different.
And I'll answer a different aspect of that is, how do you decide if this particular lab's a good place for you? Things I say, do you like the people there? Do you like the PI? Do you have the people from that lab gone on to do good things? You know, it's ideally talk to somebody who's already left the lab, who therefore has less of a stake in saying only the positive things. And are you excited by the project? So the people, the productivity, the future for those people.
Okay, as we're now approaching 3 o'clock, I'll thank you both for your time today and thank Dan for chairing the session with me. And as this is our final webinar, I'd just like to once again thank the Physiological Society for hosting this series and allowing us to discuss ideas and bring new individuals together and hopefully progress in that fashion. Thank you very much, everyone, and goodbye.